Hey there, tonight we're slipping quietly into a world often overlooked, a realm of green and hidden wonders. It's a journey into the secret lives of plants, far more dynamic than they might first appear. We'll explore their clever strategies, the ancient networks connecting forest communities, and even the secret language of their whispering roots. Prepare to discover the astonishing intelligence of the botanical world. Before we dive deeper, if you enjoy these quiet explorations, please consider liking this video and subscribing. It helps us share these scientific whispers with more curious minds. Now, let's settle in. For centuries, plants were largely seen as static backdrops to life. They stood still, rooted to the earth, seemingly passive observers. Our ancestors and even early botanists often viewed them as simple, unmoving entities. This perception of stillness is powerful. We walk past trees, admire flowers, and tend to gardens, often without truly considering the vibrant activity hidden beneath the surface. They seem to us to merely exist. But what if this stillness is a grand illusion? What if our human timescale, our fast-paced lives, prevent us from truly seeing the intricate dance unfolding in slow motion all around us? It invites a deeper, more attentive gaze. Consider a quiet forest. The trees appear motionless, the undergrowth unmoving. Yet, beneath our perception, a complex world of dynamic interactions is constantly at play. It's a hidden realm of surprising behaviors. Early botanical curiosity began by classifying and naming, but a few keen observers started to wonder about the why and how. They sensed a subtle pulse, a rhythm beyond what the naked eye could immediately discern. This quiet invitation to re-examine our assumptions led some scientists to devise new ways of seeing. What if we could speed up time, to compress days, weeks, or even months into mere moments? The results were astonishing. A vine reached out, searching for support. A seedling stretched towards the light. These slow, deliberate movements, invisible to us in real time, revealed a purposeful dance. Then there were the more immediate reactions. The Mimosa pudica, or sensitive plant, famously folds its leaves at a touch. This rapid response defied the perception of universal plant stillness. Such observations sparked deeper questions. If a plant can react to touch, what other stimuli does it sense? How does it process this information? And what mechanisms allow for such coordinated responses? Researchers began to focus on the unseen world beneath the soil. The roots, often considered mere anchors, started to reveal themselves as active, dynamic organs, constantly exploring their environment. Early studies hinted at complex interactions. It wasn't just individual roots absorbing water. There appeared to be a vast, intricate network, a hidden web of connections spanning across the forest floor. This concept challenged the isolated view of plants. What if a forest wasn't just a collection of individual trees, but a single, interconnected community, sharing resources and even information? The discovery of mycorrhizal fungi provided a crucial piece of the puzzle. These microscopic partners form symbiotic relationships with plant roots, vastly extending their reach and connecting them to others. Through these fungal networks, nutrients could be exchanged, warnings could be passed, and even support could be offered to struggling neighbors. The stillness was beginning to speak, albeit in whispers. Beyond the roots, other subtle behaviors emerged. Plants were found to track the sun, their leaves reorienting throughout the day to maximize light absorption. This was phototropism, a purposeful steering. These responses weren't random, they were precise, adaptive, and essential for survival. It suggested a form of sensory perception, a way for plants to know their environment and react accordingly. Even the timing of flowering, a seemingly simple act, revealed hidden complexity. How do plants know when to bloom? Is it merely temperature or something more intricate like counting and remembering? Through careful experimentation, scientists discovered that plants possess internal clocks sensitive to light and darkness. They can track the passage of time, a crucial ability for their life cycles. Furthermore, specific proteins and biochemical pathways were identified that act like memory banks, allowing plants to remember environmental cues such as the duration of cold periods. The stillness that once seemed so absolute began to dissolve, replaced by a picture of intricate activity and surprising intelligence. The more we looked, the more the plants revealed their secrets. We've only just scratched the surface of this botanical world. The journey ahead will delve deeper into these remarkable abilities, uncovering how plants truly thrive, adapt, and communicate. So, as you drift off, perhaps consider the quiet power of the green world around you. The stillness speaks if only we learn to listen. More wonders await in our next chapter. We've begun to uncover the hidden dynamism of plants, sensing their subtle movements and interconnected whispers. But the botanical world holds even more startling revelations, challenging our deepest assumptions about life itself. Imagine the shock centuries ago when European naturalists ventured into new lands. They encountered plants that defied all known categories, not merely absorbing nutrients from the soil, but actively capturing living creatures. It was an idea almost unthinkable. A plant, rooted and seemingly passive, could be a hunter. Early reports were met with skepticism, even disbelief. How could a plant possibly possess such predatory instincts? Yet the evidence was undeniable. From the bogs of the Carolinas came accounts of the Venus flytrap, a plant so peculiar it seemed plucked from a different realm of biology. Its hinged leaves were an enigma. Scientists, both amateur and professional, began to observe this botanical marvel. They noted its two distinct lobes resembling an open mouth, fringed with stiff, interlocking cilia. Inside, tiny hairs stood sentinel. These were the triggers the plant's sensory array. A single touch from an unsuspecting insect would not activate the trap, but two touches in rapid succession within roughly 20 seconds signaled an actual meal. This double-tap mechanism prevents false alarms, saving the plant precious energy. Once triggered, the lobes snap shut with astonishing speed, often in less than a tenth of a second, imprisoning the prey. The mechanism behind this rapid closure baffled researchers for generations. It wasn't muscle, but a complex interplay of turgor pressure. Water rapidly redistributes within specialized cells, changing the lobe's shape. Once sealed, the real work began. Glands lining the inner surface of the trap start to secrete digestive enzymes, breaking down the insect's soft tissues. It's a slow, deliberate process taking several days. Across the globe, other botanical predators were discovered. The pitcher plants, found in diverse habitats from steamy jungles to cool bogs, employed a different, yet equally ingenious, strategy. These plants form modified leaves shaped like deep, hollow pitchers. Their vibrant colors, often reds and purples, along with secreted nectar, act as irresistible lures, drawing insects closer. The rim of the pitcher is often exquisitely slippery, especially when wet. Below the rim, the inner walls are coated with a fine, waxy powder or downward-pointing hairs, making escape nearly impossible. At the bottom of the pitcher lies a pool of liquid. This isn't just rainwater, it's a carefully crafted cocktail of digestive enzymes, acids, and often symbiotic bacteria. Researchers painstakingly analyzed this fluid, identifying enzymes similar to those found in animal digestive systems. The plant wasn't just trapping, it was actively breaking down its prey to absorb vital nutrients. The diversity among pitcher plants is vast. Some are slender and upright, others squat and bulbous. Each has evolved unique adaptations to attract, trap, and digest a specific range of prey. Observing these plants in their natural habitats revealed the full extent of their deceptive brilliance. They are not merely passive receptacles but active participants in the food chain, rewriting botanical rules. The sweet nectar, strategically placed, acts as a fatal allure. Insects, drawn by the promise of a sugary feast, venture onto the treacherous rim, oblivious to the watery grave below. Through careful experimentation, scientists discovered that the plant could sense the presence of prey not just through touch, but also through chemical signals, adjusting its enzyme production accordingly. The formation of these elaborate traps is a testament to evolution's ingenuity. Each picture is a marvel of biological engineering, perfectly adapted to its predatory role. Unraveling the molecular pathways behind enzyme production and secretion became a new frontier. How does a plant, without a nervous system, orchestrate such complex biochemical processes? The discovery of carnivorous plants forced botanists to reconsider the very definition of a plant. They were not just producers, some were also consumers, blurring the lines of the natural world. These revelations fueled further expeditions, inspiring naturalists to search for more botanical anomalies. The world of plants, once thought predictable, was proving to be full of surprises. From the snapping jaws of the flytrap to the watery pits of the pitcher, these plants showcase an astonishing range of adaptive intelligence. They hunt not with speed, but with cunning, patience, and intricate design. Yet these are just the most obvious hunters. What about plants that don't wield visible traps but employ subtler, unseen methods of interaction, communication, and even defense? The botanical world's intelligence extends far beyond the dramatic capture of prey. In our next chapter, we'll delve beneath the soil, exploring the ancient, silent networks that bind entire forest communities. We've seen how some plants, like the Venus flytrap and pitcher plants, unveil their predatory nature with dramatic, visible traps. But the botanical world's ingenuity extends far beyond these obvious hunters, into realms of subtle allure and silent, swift assimilation. In the quiet laboratories and field expeditions of the 19th and early 20th centuries, as microscopy and chemical analysis advanced, scientists began to probe the ingenious biochemistry of other, less flamboyant, carnivorous species. One such marvel was the sundew, a plant whose delicate beauty belied its deadly efficiency. Its leaves were adorned with what appeared to be tiny, glistening dewdrops, sparkling like jewels in the sun. These weren't dewdrops at all, but sticky, sugar-rich mucilage secreted by specialized tentacles covering the leaf surface. This sweet, deceptive lure was the first step in its silent hunt. An unsuspecting insect, drawn by the shimmer and the promise of nectar, would land on the leaf, immediately becoming ensnared in the tenacious, glue-like secretion. Its struggles only cemented its fate. What fascinated early observers was the plant's active response. Once prey was caught, the surrounding tentacles, sometimes the entire leaf, would slowly begin to curl inward, enveloping the insect. This curling motion, though slow by human standards, was a purposeful act, ensuring maximum contact between the prey and the digestive glands on the leaf surface. It was a botanical embrace of death. Once secured, the glands would release a cocktail of digestive enzymes, breaking down the insect's soft tissues. The plant would then absorb the vital nutrients, particularly nitrogen, directly through its leaves. This process, observed through early microscopes, confirmed the sundew as an active predator, not merely a sticky trap. It was a testament to evolution's varied paths to survival. But the botanical world held even more astonishing mechanisms. Far from the bogs where sundews thrived, in quiet ponds and slow-moving streams lived the bladderworts, masters of aquatic ambush. These submerged plants possessed tiny, hollow bladders, each equipped with a delicate trapdoor and a few sensitive trigger hairs. They were almost imperceptible to the naked eye, yet incredibly sophisticated. The bladderwort created a vacuum within its trap. Water was actively pumped out of the bladder, causing its walls to flatten inwards. This created a negative pressure, a coiled spring of potential energy. When a small aquatic creature, like a water flea or a mosquito larva, brushed against the trigger hairs, it initiated an event of breathtaking speed. The trapdoor, held shut by internal pressure, would instantly snap open. The vacuum inside the bladder created a powerful suction, pulling water and the unsuspecting prey into the bladder in less than a millisecond. Once the prey was inside, the trapdoor would reseal and the bladderwort would begin the slow process of digestion, absorbing the nutrients from its captive. It was a silent, rapid, and utterly effective ambush. These remarkable adaptations, whether the sticky embrace of the sundew or the explosive suction of the bladderwort, were not random occurrences. They arose from profound evolutionary pressures. Scientists observed that carnivorous plants predominantly thrived in environments where the soil was notoriously poor in essential nutrients, particularly nitrogen and phosphorus. Bogs, fens, and acidic wetlands offered abundant water and light, but lacked the mineral wealth that most plants relied upon. Survival in these harsh conditions demanded extraordinary solutions. Carnivory, therefore, emerged as an ingenious survival strategy. By actively capturing and digesting insects or small aquatic organisms, these plants bypassed the nutrient-deficient soil altogether. They had, in essence, turned the tables, becoming active predators in a world where plants were conventionally seen as passive producers. It was a profound redefinition of their role. The intricate biochemistry behind these digestive processes, the precise timing of their traps, and the lures they employed, spoke of a deep, embedded intelligence. Understanding these silent hunters opened new avenues of inquiry. If plants could devise such complex strategies for acquiring nutrients, what other hidden abilities did they possess? The pursuit of sustenance was one driving force, but what about communication, cooperation, and the sharing of resources? Could plants also build vast, unseen networks to support entire communities? The intelligence of the botanical world, once considered limited, was expanding with every new discovery. From individual hunters, our gaze now turned to the collective wisdom of the forest. As we settle into the quiet, consider the vast, ancient networks that connect forest communities. In our next chapter, we will delve beneath the soil, exploring the secret language of whispering roots. We've seen how some plants, through dramatic traps or subtle lures, have become active hunters, challenging our understanding of the botanical world. But the intelligence of plants extends far beyond individual survival. It weaves into vast, ancient networks, binding entire forest communities. Consider the seemingly simple act of a tree growing. It stands tall, its branches reaching for the sun, its leaves converting light into energy. We see the majestic canopy, but what truly underpins its silent, enduring strength? The secret lies beneath the surface, in the hidden world of the soil. This isn't inert dirt, it's a bustling metropolis of life, a dynamic realm where countless organisms interact in ways we are only beginning to comprehend. For a long time, botanists viewed roots primarily as anchors, passive structures designed to hold the plant in place and absorb water and basic minerals. Their true complexity remained largely unseen, a mystery buried in the darkness. But a few pioneering minds, driven by an insatiable curiosity, began to look closer. They wondered what delicate structures might exist, what unseen partnerships could be forming in the dense, dark earth. In the late 19th century, a German botanist named Albert Bernhard Frank made a pivotal observation. Examining tree roots, he consistently found them intertwined with a dense network of fungal threads. This was a radical idea. Fungi were often seen as agents of decay or disease. The notion that they could form a beneficial, intimate relationship with plants was met with considerable skepticism in the scientific community. Frank, convinced of its significance, coined the term mycorrhiza, derived from the Greek words myx, fungus, and rhiza, root. It was a name that would redefine our understanding of forest ecosystems. The work was painstaking. Researchers had to carefully extract root samples, clean them without damaging the delicate fungal structures, and then prepare them for microscopic examination. It required immense patience. Under the lens, the truth became clearer. These weren't casual encounters. The fungal hyphae, the microscopic threads of the fungus, were intimately associated with the plant roots, sometimes even penetrating their cells. The question then became, what was the nature of this relationship? Was it parasitic, with the fungus benefiting at the plant's expense, or was there a deeper, more cooperative exchange at play? Through careful experimentation, the mutually beneficial nature of the symbiosis began to reveal itself. Plants, through photosynthesis, produce sugars, their primary source of energy. These sugars, rich in carbon, are essential for the plant's growth and survival. The plant, it was discovered, willingly shares a significant portion of these precious carbohydrates with its fungal partners. In return, the fungi provide an invaluable service. Their hyphae are far finer and can penetrate soil pores much smaller than plant roots can. They act as a vast, extended root system for the plant. This allows the plant to access water and crucial nutrients like phosphorus and nitrogen from a much larger volume of soil than its own roots could ever reach. The fungi are master scavengers of the earth. This discovery wasn't just about individual plants, it was about entire communities. The mycorrhizal fungi don't just connect to one plant, they form vast, intricate networks, weaving through the soil like an ancient, living web. Through this subterranean network, trees of different species, even those seemingly isolated, could be connected. It was a revelation that transformed our understanding of forest ecology. This connection allowed for remarkable acts of cooperation. Older, established trees could transfer excess sugars to struggling seedlings, offering vital support in their formative years. Beyond nutrients, researchers began to suspect that these networks could also facilitate communication. Could plants send warning signals through the fungal web, alerting neighbors to pest attacks or environmental stressors? The idea of a wood wide web began to take shape years before the term was coined. It suggested that forests operated as superorganisms, where individual trees were part of a larger, interconnected intelligence. This profound discovery fundamentally shifted our perception of forests. They were no longer just collections of individual trees, but complex, cooperative communities sustained by ancient, hidden bonds. The fragility of these networks also became apparent. Disturbances to the soil, like extensive logging or certain agricultural practices, could sever these vital connections, impacting the health of the entire forest. The research continued, revealing more nuanced aspects of mycorrhizal relationships. Different fungal species formed different types of associations, each with unique benefits and strategies. The ancient weave of mycorrhizal mysteries continues to unravel, revealing new layers of complexity and cooperation. How do these networks truly speak? What subtle signals pass between root and fungus? As we drift into sleep, consider the silent, whispering language beneath your feet. In our next chapter, we will delve deeper into the secret language of roots, exploring how plants communicate and remember. We've begun to understand the physical architecture of the mycorrhizal networks, those ancient fungal threads binding roots beneath the forest floor. But the true marvel lay not just in their existence, but in what flowed through them. The question that captivated researchers in the late 20th century was profound. If these networks connected individual trees, what exactly were they sharing? Was it merely water and basic minerals, or something far more complex? The prevailing view of forests was still one of fierce competition. Trees vied for light, water, and nutrients, each an isolated entity striving for dominance. The idea of widespread cooperation seemed almost counterintuitive. Yet a few persistent scientists, notably Dr. Suzanne Simard, began to hypothesize a different reality. She proposed that older, established trees, often called mother trees, might act as central hubs, nurturing younger saplings. This concept of a mother tree was revolutionary. It suggested a level of maternal care and resource redistribution within a forest community, challenging the purely competitive model of plant life. Proving this, however, was incredibly difficult. How could one track the movement of resources unseen through the dense, dark network beneath the soil? Traditional methods were insufficient. The breakthrough came with advancements in isotopic tracing techniques. Scientists could now introduce stable isotopes like specific forms of carbon into a donor tree and track their movement. A mother tree would be fed a small amount of carbon dioxide containing a unique isotopic signature. This labeled carbon would then be incorporated into the sugars produced through photosynthesis. The next step involved monitoring nearby seedlings and saplings. If the mother tree was indeed sharing resources, these younger plants should eventually show traces of the same labeled carbon in their tissues. The results were astonishing and compelling. The labeled carbon, initially introduced into the mother tree, began appearing in the leaves and roots of surrounding seedlings, sometimes even those of different species. This provided irrefutable evidence of sugar transfer, particularly to seedlings that were shaded or struggling. The mother tree was indeed nourishing its younger, less established neighbors through the fungal network. The implication was profound. Forests were not just battlegrounds of competition, but intricate cooperative communities. Individual trees were part of a larger, interconnected intelligence, sharing their bounty. The research expanded beyond carbon. Scientists began to trace other vital nutrients such as nitrogen and phosphorus through the same fungal pathways using similar isotopic methods. The evidence mounted. These underground networks facilitated the sharing of a wide array of resources, ensuring that the entire forest community had access to what it needed to thrive. This resource allocation was not random. It appeared to be a dynamic system, responding to the specific needs of individual plants within the network, almost as if the forest was regulating itself. But what about communication beyond nutrients? Could these networks also transmit warning signals? Could a tree under attack tell its neighbors about the impending danger? Experiments were designed to test this. One tree in a connected network would be subjected to a pest attack, such as an infestation of aphids or caterpillars. Remarkably, neighboring trees, even those not yet directly affected by the pests, began to show increased production of defense chemicals. They were preparing for an attack that hadn't reached them yet. The hypothesis formed. Chemical signals, possibly allelochemicals, were being transmitted through the mycorrhizal fungal networks, acting as an early warning system. Further research involved identifying the specific compounds involved and tracing their pathways through the fungal hyphae. The invisible language of the forest was slowly being deciphered. The term Wood Wide Web began to circulate, capturing the imagination of both scientists and the public. It was a perfect metaphor for the vast, interconnected intelligence beneath our feet. This profound understanding transformed our view of forests from mere collections of trees into complex superorganisms, where individual parts contributed to the health and resilience of the whole. The fragility of these networks also became evident. Human activities such as clear-cutting or aggressive soil disturbance could sever these vital connections, impacting the entire ecosystem's health. This shift in understanding was a testament to the power of patient observation and the application of advanced technology. It revealed a hidden world of cooperation and communication, previously unimagined. The Wood Wide Web showed us that plants do not live in isolation. But beyond these grand networks, what other secret languages do roots speak? How do they process information and even remember past events? We've journeyed through the Wood Wide Web, witnessing the vast, ancient networks that bind forest communities. But beneath these grand connections, another, more intimate language exists: the silent whispers between individual roots. For decades, roots were seen primarily as absorbers and anchors. Yet, scientists in the early 21st century began to suspect a deeper intelligence, a capacity for communication far beyond mere nutrient exchange. The challenge was immense. The soil is a black box, a dense, opaque medium. How could one observe the subtle signals exchanged in such a crowded, hidden environment? The answer lay in chemistry. Roots constantly release a complex array of compounds into the soil: sugars, acids, enzymes, and other organic molecules. These are known as exudates. Pioneering analytical techniques allowed researchers to identify and quantify these minute chemical signals. It was like finally tuning into a radio frequency previously thought to be silent. The sophistication of these signals was astonishing. They weren't just waste products, they were purposeful messages, carefully crafted chemical sentences in a subterranean symphony. Roots, it turned out, were not only speaking through these exudates, but also listening. They possessed receptors capable of detecting and interpreting the chemical whispers from other plants. A fundamental question emerged: could a plant distinguish its own roots from those of its neighbors? This self-recognition ability would be crucial for resource allocation. Experiments were devised. Plants were grown with their own roots in one chamber and the roots of a different plant, or even another part of the same plant, in an adjacent chamber. The results were clear. Plants tended to avoid extensive root growth when encountering their own roots, minimizing competition for resources within their own system. However, when encountering the roots of a stranger, another plant species, or even a genetically distinct individual of the same species, their growth patterns changed dramatically. This stranger effect indicated active sensing. Roots would often grow more vigorously or even defensively when encountering non-self roots, preparing for potential competition. Beyond recognition, roots also use chemical signals for defense. When attacked by root-feeding nematodes or fungi, a plant can release specific compounds into the soil. These compounds can either deter the pests directly or attract beneficial microorganisms that prey on the attackers, effectively calling for underground reinforcements. Roots also ask for what they need. If a plant is deficient in a specific nutrient like phosphorus, its roots can exude organic acids that help solubilize locked-up minerals in the soil. This chemical signaling allows plants to actively mine the soil, making otherwise unavailable nutrients accessible. It's a sophisticated form of nutrient seeking. Perhaps the most celebrated chemical dialogue occurs in symbiotic relationships, like that between legumes and nitrogen-fixing bacteria called rhizobia. Legume roots release specific chemical compounds called flavonoids into the soil. These flavonoids act as a unique invitation, signaling to the rhizobia that a suitable host is nearby. Upon receiving this chemical cue, the rhizobia respond by producing their own signals, which in turn trigger the plant to form root nodules, specialized structures to house the bacteria. Within these nodules, the bacteria convert atmospheric nitrogen into a form usable by the plant, while the plant provides sugars and a protected environment. A perfect chemical partnership. The discovery of these intricate root communications fundamentally changed our view of plant intelligence. It wasn't just about growth, it was about active perception and response. The soil, once considered merely a substrate, was revealed as a vibrant, chemically active medium, teeming with conversations between plants and microbes. This subterranean symphony, a constant exchange of chemical whispers, underpins the health and resilience of entire ecosystems. It is a language we are still learning to understand. The concept of roots listening and speaking through chemical exudates shows us that plants are far from passive. They are active participants in a complex, ongoing dialogue with their world. As we rest, consider these hidden conversations beneath the earth. But what about the signals above ground? In our next chapter, we'll explore how flowers count, remember, and orchestrate their astonishing blooms. We've explored the silent symphonies beneath the soil, discovering how roots communicate and share resources. But above ground, another mystery unfolded for scientists. How do plants, without eyes or brains, know when to burst into bloom, seemingly tracking the precise rhythm of the seasons? For centuries, farmers and gardeners observed this precise timing. Lilacs in spring, chrysanthemums in autumn. It seemed intrinsically tied to the yearly cycle, yet the exact trigger, the plant's internal signal, remained elusive to scientific understanding. Early theories often pointed to temperature as the primary cue, the warmth of spring, the chill of autumn. Yet, researchers noted that plants sometimes bloomed out of sync with temperature shifts, suggesting another factor was at play. In the early 20th century, two American scientists, W. W. Garner and H. A. Allard, stumbled upon a peculiar tobacco mutant called Maryland mammoth. It grew enormous leaves, perfect for cultivation, but stubbornly refused to flower during the long summer days. They moved the giant tobacco plants into a greenhouse, experimenting with various conditions. Only when the natural days grew shorter, or when they artificially shortened the light period by covering the plants, did the Maryland mammoth finally bloom. This groundbreaking observation led Garner and Allard to a new concept: photoperiodism. Plants, it seemed, weren't just sensing the seasons, they were precisely measuring the length of day and night, using it as a critical cue for their life cycles. They identified short-day plants like chrysanthemums and coffee, which flower when the nights are long, and long-day plants such as spinach and barley, needing short nights to trigger their bloom. Further meticulous experiments revealed a crucial detail. It wasn't the length of the *day* that was most critical, but the continuous, uninterrupted period of *darkness* that truly regulated flowering. A brief flash of light in the middle of the night, even for a few minutes, could effectively trick a short-day plant, preventing it from flowering. The darkness for these plants had to be complete and sustained. This led to a profound question in botanical research. How did a plant see light and darkness, and how did it measure time so precisely? What internal sensor, what biological mechanism was at play? Early experiments showed that removing the leaves from a plant prevented it from flowering, even if the rest of the plant received the correct light cues. This suggested the leaves were the primary light receptors, the plant's eyes for sensing time. The idea of a mobile flowering signal emerged. Scientists hypothesized a hypothetical hormone dubbed florigen, which was thought to travel from the light-sensing leaves to the buds, triggering the astonishing bloom. Decades later, in the 1950s, a significant breakthrough occurred. Scientists at the USDA finally isolated a specific light-sensitive pigment responsible for this process: phytochrome. Phytochrome existed in two interconvertible forms: PR, which preferentially absorbs red light, and PFR, which absorbs far red light. It functioned as a molecular switch, responding to the quality of ambient light. During daylight, the abundance of red light rapidly converts most of the PR form into the PFR form. PFR is considered the active form, signaling the presence of light to the plant's internal systems. In darkness, the PFR form gradually reverts back to PR. This slow conversion, a steady decline of the active form, acts as the plant's internal clock, precisely measuring the length of the night. For short-day plants, a long, unbroken night allows enough PFR to convert back to PR. When the PFR level drops below a certain critical threshold, the signal to flower is finally initiated. For long-day plants, the opposite is true. A short night means that PFR levels remain high enough throughout the dark period, signaling that conditions are right for their specific blooming cycle. This also explained the puzzling flash of light phenomenon. A brief exposure to red light during the night converts PR back to PFR, resetting the internal clock and preventing flowering in short-day plants. While phytochrome was a major discovery, the plant's timekeeping mechanism is far more complex. It intricately interacts with an internal circadian rhythm, a daily biological clock, to fine-tune its responses to environmental cues. This sophisticated light-sensing ability isn't just for flowering. It also controls other crucial seasonal behaviors, such as the breaking of bud dormancy in spring or the programmed shedding of leaves in autumn. This brings us back to the idea of plant memory. Beyond light, some plants require a specific period of cold, a process called vernalization, to remember winter and trigger flowering in the subsequent spring. The plant remembers this cold exposure through subtle, long-lasting changes to its gene expression. These epigenetic modifications don't alter the DNA sequence itself, but rather how its genes are read and activated. These discoveries revealed plants as astonishingly sophisticated timekeepers, able to count sunlight, measure darkness with precision, and even recall past environmental conditions over long periods. The flowering clock is just one facet of botanical intelligence, showing a precise, internal awareness of their world. But how do these internal states translate into broader, coordinated actions, and what other astonishing abilities lie hidden within the botanical realm? We've seen how plants precisely count sunlight, orchestrating their blooms with an internal clock. Yet, for many species, light alone isn't enough. They possess an even deeper form of recall, a botanical memory that spans weeks or even months, ensuring they flower at precisely the right moment after winter's chill. Consider winter wheat. Planted in autumn, it endures the cold months, seemingly dormant. But without that sustained period of low temperatures, it simply won't produce grain in the spring. This phenomenon, known as vernalization, puzzled scientists for decades. Farmers and botanists had long observed this requirement. It was clear that the cold wasn't just pausing growth, it was actively priming the plant for future flowering. The question was, how did the plant remember this cold exposure once spring arrived? Early experiments involved exposing seedlings to controlled cold temperatures, then moving them to warmer conditions. Researchers meticulously tracked their development, trying to pinpoint the exact duration and intensity of cold needed. The challenge was to separate the memory from the immediate effects of cold. Was it a simple on/off switch, or a more nuanced accumulation of cold units? The answer lay at the molecular level, hidden within the plant's genetic machinery. Scientists began to suspect that the cold wasn't just affecting existing proteins, but fundamentally altering how certain genes were expressed. It was a change in the plant's internal programming, a long-term adjustment. The concept of epigenetics, the study of heritable changes in gene expression that do not involve changes to the underlying DNA sequence, offered a powerful new lens through which to view plant memory. Imagine your DNA as a vast instruction manual. Epigenetic marks are like sticky notes or bookmarks telling the cell which pages to read and when. They don't change the words on the page, but they control access to them. For vernalization, a key player emerged, a gene called FLC, or flowering locus C. In many plants, FLC acts as a repressor, actively preventing flowering. It's like a brake on the flowering process. During the cold period, the plant begins to gradually silence this FLC gene. It doesn't remove the gene, but it adds epigenetic marks that make it harder for the cell to read or activate FLC. The longer the cold exposure, the more effectively FLC is silenced. This silencing is a cumulative process, a slow accumulation of memory of winter's duration. Once the cold period is sufficient and FLC is sufficiently silenced, the brake on flowering is released. Even when temperatures rise again, this epigenetic mark remains, a persistent memory of winter. This stable epigenetic memory ensures that the plant doesn't flower too early during a fall spring, but waits until the true warmth of the season has arrived, maximizing its chances of successful reproduction. The sophistication of this molecular memory is profound. It demonstrates that plants can not only sense their environment but also integrate information over time, storing it for future use. Different plant varieties have evolved different FLC versions and different sensitivities to cold, allowing them to adapt to specific climates and ensure their flowering aligns with local conditions. This flexibility in memory is crucial. Plants can, over generations, adjust their vernalization requirements, subtly shifting their flowering times in response to changing environmental pressures. The molecular tools used to uncover these mechanisms are incredibly precise, allowing scientists to observe changes at the level of individual genes and their regulatory marks. This research reveals how environmental cues like cold can leave lasting imprints on a plant's genetic expression without altering its fundamental DNA code. It's a dynamic, adaptive system. The beauty of a spring bloom, therefore, is not just a response to warmth, but the culmination of a long, silent calculation, a deep memory of the winter that has passed. As climates shift, understanding this vernalization memory becomes increasingly important. Milder winters can confuse plants, leading to erratic flowering or reduced yields in agriculture. Botanists are now studying how plants might adapt or fail to adapt their flowering times in response to these rapid changes, highlighting the flexibility and limits of their epigenetic memory. The flowering clock and its memory of cold reveal a plant's astonishing capacity to process complex environmental information and orchestrate its most vital life stages with precision. This deep dive into how plants remember seasons underscores the quiet intelligence permeating the botanical world, a far cry from the passive entities once imagined. From counting sunlight to recalling the cold, plants are master timekeepers and record keepers. Their internal world is far richer and more dynamic than we often perceive. As we settle into the quiet, consider the silent, meticulous work of plants, remembering seasons, adapting to change. But what about their ability to truly learn from experience, to form habits, and even solve problems? More wonders await. We began this journey by challenging the stillness of plants, revealing their surprising dynamism. Remember the carnivorous plants, those silent hunters? Their ingenious traps, like the snapping jaws of the Venus flytrap or the slippery pits of the pitcher plant, were not mere accidents of nature. They represented active problem-solving, an evolutionary response to nutrient-poor soils. The sundew's sticky embrace, the bladderwort's explosive suction, each a testament to a complex, adaptive design perfected over millennia. Then we delved beneath the soil, discovering the ancient weave of mycorrhizal networks. This wood-wide web revealed a hidden world of cooperation, where trees shared resources and even warnings through fungal intermediaries. It reshaped our understanding of forests, transforming them from competitive battlegrounds into integrated communities. The exchange of carbon, nitrogen, and phosphorus spoke of a collective intelligence, a shared destiny. Beyond these grand networks, we heard the subtle whispers of individual roots. Plants communicate through chemical exudates, recognizing kin, deterring pests, and even signaling their nutrient needs to microbial partners. And above ground, the flowering clock revealed an astonishing mastery of time. Plants count sunlight, measure the darkness, and orchestrate their blooms with breathtaking precision, using internal biological clocks and light-sensitive pigments like phytochrome. This led us to the concept of plant memory. The quiet intelligence of vernalization showed plants remembering winter's chill through subtle, long-lasting epigenetic changes to their gene expression. They don't just react, they integrate information over time, storing it for future use, ensuring they flower at the optimal moment. This is sophisticated information processing, far beyond simple reflexes. All these observations, trap design, communication networks, memory, problem-solving, have led to a profound debate in the scientific community. Can we call this intelligence? The traditional definition of intelligence often centers on brains, nervous systems, and conscious thought, attributes absent in plants. This makes the conversation complex, challenging our anthropocentric views. Some argue that terms like cognition or intelligence are too loaded, implying animal-like consciousness. They prefer adaptive behavior or complex information processing, emphasizing the plant's biological mechanisms. Yet others contend that intelligence should be defined by outcomes. The ability to sense, integrate information, learn from experience, and adapt to changing environments to ensure survival and reproduction. From this perspective, plants undeniably exhibit intelligent behaviors. They solve problems, optimize resource acquisition, defend themselves, and communicate with remarkable sophistication. The emerging field of plant neurobiology, though controversial due to the neuro part, seeks to understand how plants coordinate these complex behaviors without a central nervous system. It focuses on the intricate networks of signaling molecules, hormones, and electrical impulses that allow plants to process information and make decisions at a cellular and systemic level. Regardless of the terminology, the research has fundamentally altered our perception of the botanical world. Plants are not passive, silent entities, but active, dynamic beings. This new botanical view fosters a deeper respect and wonder. It invites us to see every tree, every flower, every blade of grass not as a static backdrop, but as a participant in a vast, interconnected web of life. It reminds us of the quiet complexity that surrounds us, a complexity often overlooked in our fast-paced human world. The plant kingdom holds secrets we are only just beginning to decipher. The patience required to observe, the meticulousness to experiment, and the openness to revise our assumptions are the hallmarks of this scientific journey. Each discovery, from the snapping trap to the whispering root, has been a testament to human curiosity and the endless pursuit of understanding. The botanical world, in its quiet, enduring way, continues to surprise us, challenging our preconceived notions of life and intelligence. It encourages us to listen more closely to the silent conversations, to observe with greater attention, and to appreciate the profound wisdom embedded in the green world. The mystery doesn't vanish, it deepens. For every answer we find, new questions emerge, inviting us to continue our quiet exploration. The intelligence of the botanical world is not loud or flashy, but subtle, persistent, and deeply integrated with the rhythms of the earth. It is a quiet symphony that plays on whether we are listening or not. And so, as the moon climbs high, casting its gentle glow upon the silent, sentient green, sleep well, knowing the world around you is alive with wonders, quietly unfolding its secrets, waiting for your return.